Hey everybody, and welcome to chapter number 4. We're going to be talking about visual object recognition in this chapter, and if you haven't watched the chapter 3 video or read that chapter in the textbook, I would strongly recommend it because that chapter talks a lot about the early visual system, and we're going to be building upon a lot of those ideas. So if you don't watch that, whenever you hear me talk about the what versus where pathways, or if you hear me talk about top-down versus bottom-up processing, I want to make sure that we're on the same page. So check that one out if you haven't checked this one. If you have checked that one out, then let's go. Let's do this. So today we're going to be talking about visual feature net models, which are sometimes referred to as the cognitive demons. And I know that that is not nearly as cool as it's going to sound. But we're going to be talking about that. We're going to be talking about the feature integration theory from Ann Treisman and Gilad from 1980. We're also going to be talking about the recognition by components model from Irving Biederman to talk about how we put features together and recognize them as objects, and then we're going to wrap things up by talking about face perception. And speaking of face perception, the videos that I have for you for this chapter are largely going to be prosopagnosia related. If you're not familiar with prosopagnosia, it is also referred to as face blindness. This is what we're going to be talking about at the very end of this chapter, but if you want to get a quick look at what prosopagnosia looks like, then you can check out any of these videos that kind of walk through it. I would recommend, I think if you're looking for a place to start, the newer one right here. This is a good one to start with, and then if you like it, then check out the longer video. But basically the thought experiment is this: what if you could recognize everything, no problem? You can recognize what a mug is, what a microphone is, what a phone is, but you can't recognize people's faces. So if my wife were to walk in, I would not know who that was. And I might be able to guess it based on hair and based on her outfit, because I've seen that earlier today. But I would not recognize her just by her face. And if you're wondering what might that look like, check out these videos, and you can see people who live with that condition, which is called prosopagnosia. While we are on the subject of agnosias, I wanted to mention this image because I didn't last chapter, and I should have. When we were talking about visual, or maybe I did actually, I think I did. I record these really late, y'all, because when I try to record during the day, I don't know, the vibes are never right. I like at night because I know I'm not going to get interrupted, and I get a little bit loopy, and I feel like that can be helpful for making this not extremely boring to listen to. Hit that like and subscribe button, y'all. But I wanted to bring this up one more time because we're talking about visual object recognition. But if you have visual agnosia, you may remember from last chapter, that's whenever you damage the inferotemporal cortex, which is part of that ventral stream of the what pathway. That if you damage the inferotemporal cortex, then you can see where things are. You can see roughly their shapes and roughly their colors, but you can't actually say what it is that they are looking at. So if I was to try to describe this to you, I can't really put it into words. I would say it looks like, I don't know, like a toucan with hair, right? So it looks like a bird, with a furry bird or something. And that's about as descriptive as maybe somebody that has visual agnosia would get. In terms of their ability to describe what they see, because their brain's not able to put together the objects, and this specific image has usually been passed around as a demonstration of that. People sometimes say that this is an approximation of what it's like to have a stroke, and usually whenever people say that, what they're really saying is this is what it would be like to have visual agnosia, because a lot of visual agnosia patients begin because of pinpoint strokes in specific parts of their brain. All right, I just wanted to reshare that with you to get us thinking about that as we talk about object recognition. So as we talk about that, let's talk about how we study it. I wanted to walk through a visual search demo. This is going to be kind of similar to the one that we did, the Sternberg search task that we did in chapter 1, but I wanted to go over it another time to kind of, again, get us thinking about the ways that cognitive psychologists study the mind and study the brain. And usually it's with tasks that are maybe a little bit more simple than you thought. It's just that the way that we manipulate these independent variables to try to make inferences about the internal mechanisms are a little bit out of the box. Maybe that's where we're being creative. Usually in the experimental tasks, they're pretty straightforward and they can be a little bit simple, I guess. So here's an example of a simple one. This is a visual search task. So your job would be to find the vertical line. So if you were doing this in a laboratory, you'd probably have a keyboard and you press one button if you see the vertical line, and you press another button if you don't. And so the question is of accuracy and of reaction time. So how quickly can you respond and how many times can you get it right? And almost always accuracy is really, really high because, again, these are really easy. So for example, do you see the vertical line? Yes, it popped out at you, right? So this right here, it pops out at you because why? Because it's not like any of these other ones, right? Let's move on. Find the green colored line. And again, this one pops out at you, right? So the point that these two really simple experiments are making is that whenever it comes to what the target is, so what it is that we are looking for, it doesn't really matter how complex the target is. What matters is how complex the background is, or the quote-unquote noise of the environment. So in this case, the vertical line shares only really one property with these others, and that's color, right? So it's black like all the others, but it's vertical, and it's the only vertical one, and so it kind of pops out at us. Same with the green colored line. It is the only one that is green. All the others are vertical, though, so we can rule out verticality as, you know, the thing that makes something pop out at us, because all of these are vertical, and yet the green one is one that popped out. Why? Because it has something that it does not share with the background, with the distractors. So what about this one? So let's find a vertical line that is red color. So it is standing up and it is red. This one takes a little bit more time, but not very much, you know, but maybe just a couple of split second, you know, like maybe 20 milliseconds longer, but you probably felt that little delay, right? You felt that little slowing down. That change in your reaction time is going to be important. In cognitive psychology, that difference in reaction time, even if it is as short as 20 milliseconds, is informative to us. It tells us something about the internal mechanisms of the mind. It lets us know that maybe that distinction, that choice that we just had to make was a little bit trickier than the previous ones. The previous ones we had instant saying like, yes, I know where that target is. This one took a little bit longer. Why is that? Well, this time we don't have a single unique characteristic that makes it pop out, because all of the qualities of this specific target are shared with the distractors. We have some that are red and we have some that are vertical. Even though we only have one that is red and vertical, because we have some of those shared characteristics, it takes us a little bit longer to find that specific item. So the point of all of this is that that last test was a little bit more difficult, because now we're not just searching for a feature. This time we are searching for a combination of features. We're not just searching for, is it green? We're not just searching for, is it vertical? We're searching for a stimulus that satisfies those two conditions now. So it's not just, do we satisfy this one condition? It's, do we satisfy these other conditions as well? These two conditions whenever the backgrounds share some of those attributes. So the lesson here is that the more features a target has in common with its distractors in the background, with its environment, the harder or the longer it is for us to say that we see it. We see this applied in the real world whenever we talk about things like Where's Waldo, if you remember this. I don't know if y'all, depending on how old you were, I don't know if you got these a lot growing up. For me, I was a kid in a pre-tablet, pre-smartphone world, so we didn't have screen time. Whenever our parents got bored of us asking questions, they would hand us one of these Where's Waldo books, and the whole point of it is that you're supposed to find this jerk named Waldo. Waldo wears white and red striped shirts. He wears a little beanie like he's in New England, and he's got a swooped hair that's brown, and he's got some Coke bottle glasses. If you don't see him, he is right here. I'm going to circle him right there. He's right there in the middle. This one was kind of an easy one for that reason. But the reason why this task is hard is not because Where's Waldo sticks out, right? Because he should stick out. He's wearing white and red, like if you're trying to stand out in public, it's harder to find a more loud outfit than Waldo, right? He's a real baddie. He's, trying to stand out at the club. But the issue is that here at the club, everybody else seems to be wearing red and white or a combination of red and white. So we have a lot of distractors that share these features, and for that reason, he no longer pops out. So it's not just how rich and how detailed and how complex the target is that makes it salient. It is how many features it shares with the other competing distractors in our environment. All right, so that's just a typical kind of experiment, that you might see in cognitive psychology. And again, what we're doing there is we are manipulating those independent variables. We're seeing how long it takes, in this case, reaction time. And then we're drawing inferences about that reaction time to tell us a little bit more about how our brains process the world around us and the world inside of us, if you want to think about those internal processes. So let's get into it. When we're talking about what's going on, both at a neurological level and at a theoretical level, I think a good place to start is with feature nets. Feature nets are a conceptual model here. You'll see this referred in the textbook and in other, or in most textbooks, as what is it called? The pandemonium model. And the pandemonium model talks, I don't know why they did this. Actually, I think I do know why. But basically, they refer to our object recognition processes as being handled by demons. And they mean demon in, like this way, basically. DAEMON, the same way that like your email service provider means it, like if you accidentally tried to send an email to an address that doesn't exist and you get that email back and it says something about like email server demon could not deliver it, and then a chill rolls up your spine because you're like, did I just do witchcraft because this email is demonic or something? No, basically this demon in this case is referring to like an imp, like a little helper, so that's what we're not doing any kind of satanic work here in cognitive psychology this time. But the pandemonium model refers to these demons as some of these demons are going to be detecting and handling really, really small features. So by features, I'm talking about angles of lines, curves of lines, and if that line is vertical, if it's horizontal, and so on. So basically looking at the very bare basics of what a visual feature can be. And then from there, it gets passed up, that information gets passed up to a secondary demon. That secondary demon, in this case, might be the one in charge of recognizing letters. So whenever this feature demon down here is going to pass up all of these features that it has collected, this letter demon is then going to look at in this case, let's look at the clock, let's look at the C of clock right here. So if this demon right here, it gets this bundle, these three different features, a line that's curved that way, a line that's curved that way, and a line that's curved this way. It's going to put those together and say like, oh, by Scott, that is a C. I recognize that as a C. And then from there, this demon is going to pass up to a decision demon, in this case a word demon up here, that is then going to put those together and decide on what it is recognizing. So in this case, this demon is going to be getting, all right, I got a C, I got an L, I got an O, a C, and a K, and that spells clock, and we recognize what that is, and we understand it, and then we can move on to the next word. So all of this sounds like a lot of work, but as you know, this happens almost instantaneously within about 150 milliseconds. It happens extremely quickly. But this model, what I was telling you here was, this metaphorical model with the demons. That's like a way to conceptualize it. That's a conceptual model. It's kind of like a metaphor for how this works. But this is essentially how information is processed in the cortex, in the cerebral cortex, where all of this mostly happening in the occipital lobe, that we have layers of the primary visual cortex that are doing all of these things. Where we have the V1, you may remember we had some of those receptive fields that were responding to line angles, right? So we know that there are some cells that are responding to specific line angles. Those cells are going to communicate with these hierarchical, larger, or I say larger, superordinate, I should say. I'm going to write that down because that's a big word that sounds important. Like a superordinate cell cluster that is essentially kind of going to respond if it and fire more frequently if it gets activation from cells that communicate this combination of lines. Then that network of cells, which represent the letter C, is then going to fire over to probably the lingual gyrus or somewhere like that, somewhere kind of on the border of the occipital lobe and the temporal lobe. But basically what I mean to say is that this kind of fun little process that we talked about with the demons is a metaphor, but that is essentially what is happening in the cerebral cortex whenever we are recognizing words. So it can also be, and this is a very bottom-up process that we're talking about here, is very data-driven, right? We're talking about what are we visually experiencing. So we're talking about, the visual processes here. Now, if we were to take this with a top-down approach, remember that top-down processing means that we have some expectation, we have some kind of knowledge about what to expect from this experience or from this stimulus. And we see this all the time with language, because with language, we are constantly predicting what the next word in a series of other words is going to be. Like, so if I'm telling you a story and something unexpected happens, it grabs your attention and you're like, wait a second, what? And that's partly because you're trying to guess where the story is going while it's happening in real time. And that is an example of top-down regulation, top-down processing, because your expectation for the English language is going to change what words you think are going to come next. Yes, you guessed it, because you use top-down processing to do that. So sometimes if we have features that overlap with one another in words, so in this case, let's say that we got this array, CQRN. That is not a word in English, right? It doesn't have a vowel or anything like that, but it looks like the word corn, kind of. It is totally possible that whenever we have these features that are being passed up in our feature net, that we do essentially a probabilistic analysis. And it's not conscious, but it is one of those things that, our brain is really good at doing. It's really good at finding patterns, especially patterns that we think are familiar to us. And so this word we might see for a moment as the word corn, and we recognize it as corn. Now, if you gave this same thing to somebody who does not speak a single word of English, and you asked them to copy this word down, and you flashed it in front of them, they'd probably do CQRN. If we did the exact same experiment where we flashed on a screen in front of somebody who does speak English, they'd probably say CORN. The reason for that is probability. That the English speaker is going to see that flash, and they are going to put together, oh, they look like a C, an O, an R, and an N, because in most cases, Q is not the second letter of a word whenever C is the first letter of the word. I can't think of any words that exist like that. So probabilistically, not likely. But CO we can think of lots of examples of words like that, like cone, corp, core, corn, again. And so for that reason, we're more likely to use that top-down processing, our expectation for what that word could be, to guide what we reported seeing. So just like when we were talking about this in the previous chapter, that we're more likely to see this as the word the, T-H-E, and we're more likely to see this as the word C-A-T, cat, because this ambiguous symbol is interpreted as an H here, because it's more probabilistically, is more probable that it will be. And here we perceive this as an A because it's more probable that it's an A, given the context of it. All right, so let's shift up a little bit to talk about how these features are put together. And again, this is going to kind of touch on one of the things that we talked about in Chapter 3, which is Anne Treisman's 1980 paper. I mentioned already, I love this paper. This paper rules. I would have assigned it for our class if I thought that more than 2 people would have read it. I don't think anybody would have really read it because it is very long. It's got like 12 different experiments inside of it. And that's how you know Anne Treisman. I'm gonna give her some respect and write out her name right here. She's no longer with us, but that's how you know she's a real one. Because sometimes you have researchers who are out there and they're like, hey, this university expects me to publish 4 papers a year, and I've only published 1. So what I'm going to do is for this next paper, instead of publishing it as 1 paper, I'm going to break it up into like smaller experiments and try to publish each one of them separately. That's how some researchers do it. Anne Treisman, not a chance. She's like, and that's why I love this article so much, is because it's a really good example of somebody following a hypothesis and incrementing their experimental design step by step to respond to their conclusions. So basically, she was interested in the feature binding problem. So you may remember this from last chapter. We were talking about the binding problem and about how we have this ventral stream that goes underneath the brain that responds to what something is, its identity. And then we have the dorsal stream, which is like where something is. And so we have information processed in 2 independent streams, and yet we feel like everything is one whole coherent experience in our consciousness. And so she was curious, well, why is that? And so she developed a series of tests that eventually ruled out other alternative explanations until you're left with what we're about to see here. Which, for her, the idea of attention, partly because she was attention, like that's kind of like her main gig was attention at first. And then she kind of got into this more perceptual world. But she would basically go on to show that without attention applied, visual information gets very, very messy and easy for us to get wrong. So I'm going to show you an example of what she did to show you how we know this and why we know this. So before I do that, I want to tell you about illusory conjunctions or conjunction errors. This is an error that we make whenever we see something out the corner of our eye, and you think you see something, and you like do a double take, and then when you actually look at it, it's not what you thought it was. And you can probably think of a million examples of these. I experience it a lot whenever I'm reading. So like if I'm reading and I see like out of the corner of my eye the word Western Field, or the words Western Fields, I get all excited because I'm like, oh, it says Westfield. And then whenever I turn my attention to it, then I realize it's not Westfield, it's Western Field. And it's way less interesting at that point, right? But you've probably also done this where like you think you see a friend, or you think you see a famous person out of the corner of your eye. You turn to look at it and they're not there. Or maybe there's like a shadow and you think it's a person, right? And then you turn and you look and it's not what you thought that it was. Basically, once you apply your full attention to that, then your perceptual processes are able to kind of glue that together, and we're able to recognize it for what it is. Attention going to come up a lot next chapter. Chapter 5 is all about attention, which is why this chapter is exactly where it is. The borderline between perception and attention. That wasn't a mistake. So here is an example of what Anne Treisman did to look at illusory conjunctions, these kinds of errors that we make. So just focus right here on the fixation cue. And now is as good a time as any to talk a little bit about what a fixation cue is. A fixation cue is used a lot in cognitive psychology and in research like this because reaction time is really important. We use reaction time to draw inferences about what's going on in the brain and the processes that are at play. And so it's really important for people to have the same starting point. So in this case, where we are interested in reaction time, and we're interested in looking at attention, we want to make sure that everyone's attention is at the same place at the beginning of the trial. So we want everybody to look right here at this fixation cue. So that's what that's for. At the beginning of a trial, that may people we hope at least that people are focusing at this this fixation cue at the beginning of the trial, so that if there is any kind of differences or anything interesting going on with accuracy or reaction time, that we can rule out that, you know, well, I tend to start looking over here, and Joe, he normally starts looking over here, and Susan, whenever she, you know, is left to her own devices, she starts looking for stuff over here. This rules all that out, so that way we're all looking at the same place. Just wanted to make sure that was clear. So, yeah, I want you to tell me what are the objects in the slide that is coming up. So here we go. Focus on the fixation cue. All right, here we go. Trial 1. Okay, so you probably saw something right here immediately to the left of the crosshair. And actually, I apologize for this first one because I put the key stimuli up here in the top right corner, and you probably can't see that because of my big head over here. My bad. Sorry, I should have thought about that whenever I was making this. So let me back up. Usually whenever people are doing this, if you're following along with the lecture slides that I have posted, generally people are going to look at this crosshairs, have a big distractor image next to the crosshair, and then after that we have the actual stimuli. And if you can't see it over here, it is a yellow triangle and a red square. Now whenever people do this task, they are very likely, if their attention is over here, to get this wrong. And whenever they are asked what do they see, they'll say, oh, I saw a house, or they'll say, like, I saw a red square, or a red triangle and a yellow square, where they kind of mix up the colors. Or they might say that they think that they saw like a trapezoid instead of two separate shapes. The point being that in that short amount of time, even though they were able to perceive that they saw something, they will get the characteristics of it incorrect. And that's because they don't have enough time to apply their attention to glue those features together. So here is another example. Let's do trial number 2 now. Here we go. Fixation cue. Okay, so again, it happened really quickly. I know, but that's how these experiments are. It happens really fast. You saw something grab your attention over here, and then as you did, something popped up in the bottom left-hand corner. But did you catch what it was? Let's back up. So go ahead and write down what you think that it was before you see what the answer really was. So we had this distractor come over here on the right side of the visual field, and then over in the bottom left, we had a black arrow pointing down and a blue heart. And again, whenever people do this task, because their attention is over here, they're more likely to get this wrong and say what's wrong about it. If I told people to look at this part of the screen the entire time, so I want you to look at the bottom left, you can see how easy it is to recognize those objects whenever your attention is over there. So let's take a look at it. I'm putting my cursor over there so you know where to look. Right? So when your attention is there and it's flashed in front of you, it's not hard at all. It's only hard whenever our attention is not there. Here we go. Let's do one more. Okay, so in this case, we saw something big right here, right, that grabbed our attention, and then something kind of over here, but because it was kind of close to that distractor, we're going to have people getting some of those more correct than whenever it was really far outside of our attentional field. And so in this case, people will report they saw something purple and yellow. They might be able to say exactly what the yellow thing is because that's kind of an ambiguous object. It's a plus sign, maybe, or a cross. But look at the purple moon, correct. Trial 4, here we go, you know the drill. All right, that one was really tough because we were using words, but you can also use this task with words, and what we find is that whenever we see something like this, and you have the word newt in purse, and you have a distractor grabbing that attention, that people will get this wrong, but we have conjunction errors where people will put them together to say they thought they saw the word nurse, right? Which is just a combination of those two, but if you're focused over here to begin with, your attention is already pitched over there, then in that split second, you see exactly what it is, and it's not hard at all. So basically, whenever those things appear close together in our visual field, we make mistakes only when our attention is not applied over there. So before we apply our attention, those features are not bound together, and for that reason, they become kind of difficult. So that was Anne Treisman's feature integration theory that essentially helped solve the binding problem. And you may remember that last chapter we talked about how the important thing was spatial synchrony and neural synchrony, the idea that these things happen together at the same time. And then this also showing that attention matters, that it really depends on where your attention is about how well you can put those items together. All right, now to shift gears just a little bit. We're not going to shift gears too much, though, because we're still talking research. We're going to talk about recognition by components here. So recognition by components is also another gigantic article, extremely influential, not just in the field of psychology, but also in the field of artificial intelligence and computer science, because a lot of how machines are programmed to recognize objects is pulled from this research as well. So this was published in the late 80s, so it's pretty old, but not super old. It's a little bit older than me, but not very much older. So hey, maybe my parents read this to me as a bedtime story when I was growing up, and maybe that's why I love psychology. I know that's not true because my parents are first generation, I'm a first generation graduate, and so they didn't have these kind of articles just like hanging out around the house. I was reading Cinderella. That was my favorite Disney book, and I liked it so much I had 2 copies of it. Anyways, let's get into recognition by components. Shout out to my Cinderella fans out there, though. What can I say about this article? Like I mentioned, it's a huge article, and it has, also, like Anne Treisman's article, a ton of studies in it. I want to say maybe more than 12. For some reason, I'm thinking that it might have 20 different experiments inside it. I might be getting that wrong. It certainly has 20 separate images and figures in it, which blows my mind that this journal, Psychological Review, let them get away with printing that many there. But let's get into it. So Biederman's his whole theory is famous for this notion of geons. So I'm going to go ahead and put that little bug in your head now, this notion of what a geon is. A geon is a sub-object. A geon is the smallest basic shape that you can make. If you take a complicated shape and you boil it down to its very basics, and you can't reduce those shapes into anything more simple, then that's what you're left with. That's the geons. So his theory about how vision is processed, or how we recognize objects, and again, this is a theory, but this one has a lot of really good evidence to back it up, both neurological and behavioral. So sometimes we get kind of caught up in this is still a theory, because it's really hard to prove, because it is really kind of hard to be 100% dead certain about some of these things because it would require unethical human testing to get to that certainty, where we would need to like pop open someone's brain and just start like fiddling around in there, you know. So we can't do that, and so that's why some of these things like recognition by components remains like a theory, because it can't be a law until we like violate the Geneva Convention, essentially. All right, anyways, there are 3 stages of this theory. The first thing when we see a complex object is that we're going to break it down into its sub-objects. And that's what you're seeing here. So basically, if we were looking at this lampshade, right? This lampshade, very easy. You can break it up into 2 separate objects where you have like this funnel and then you have the cylinder. So you got these 2 basic sub-objects, these 2 geons. Same thing with this suitcase, right? The suitcase has like this loop and also has this rectangular prism, this big rectangle. I really like these 2 examples right here. So this is a mug and this is a bucket, right? So they both have the same type of geons. They both have a cylinder and they both have this loop. But based on the placement and the relationship of these geons, we know which is which. We know that this is a cup and we know that this is a bucket. And we know that because the handle or the loop is on the side of the cylinder here and is on top of the cylinder here. And we recognize that pattern, and that's how we know which is which. That's essentially Biederman's argument about how we process how we process objects in a nutshell. So we can apply the same thing for even more complicated things, like you see over here, these living organisms, where like check out this horse. So if you take a horse and you break it down into its sub-objects, it's basically just an array of cylinders, right? We have like 4 long cylinders here that represent the legs. We got a long torso, we got the neck, and we got the face, we got the tail. The interesting thing, though, is that, you know, if you didn't see that this said horse down here, and I just asked you to guess what it was, if you said it was a dog, I don't think anybody would laugh at you, right? Like, because it does kind of look like a dog, because dogs and horses have essentially the same kinds of geons. The only difference, and they're kind of in the same array, the same layout, the difference is really how big they are, right? And that tells us which one's a horse and which one is a dog. But looking at these 2, we see that they also have the same geons, right? They have the same geons. They're in the same place. The only thing that's different is the relationship that they have to each other in terms of their size. So we see that this one has a really long geon right here. This cylinder is very, very long compared to the rest of it. And then we're like, oh, yeah, that's a giraffe, right? We know that's a giraffe because the only case where we see geons in this array with a really long geon right here is when we're looking at a giraffe. And you can apply the same logic, the same kind of mental exercise to these subclasses, right? So we're looking at humans versus apes. So we're both primates. So if I were to ask you, you know, like this right here, like what is this right here? If you said a human, I don't think anybody would laugh at you, right? Because it's very close to being a human. But when we compare these 2, we can see the differences, that basically humans have really long geons down here, whereas apes not so much, and that the geons right here are short for humans and are long for apes, and that is essentially the difference. So we can recognize what these things are without having to look at the color, without having to look at the texture of what it is, that essentially our brain is going to start making guesses about what we see at this early stage, whenever it is putting together these basic shapes to create more complex shapes. So the 3 stages that we have here, the first is that we're going to be taking this object and we're going to break it down into its sub-objects. That's what we did here. The next step, step number 2, is that we're going to take those geons and we are going to classify them into known categories of those geons. So, for example, if we see something like this as number 3, we know that's a cylinder. If we see something like this, okay, that's a cone and a pyramid and a horn and a football and a wine glass. This is a loop and a funnel and a rectangle. You get the idea, that there are certain geons that are repeated, that we are familiar with, that we see a lot. Apparently there are 36 different kinds of geons. I'm not going to ask that you know which is which, and even for an activity that I have for this specific chapter is for you to pick out at least 2 complex objects that are related, just like we see here, where like a horse and a giraffe are very similar, and then you have to identify what the geons are that make up that specific complex object. And then the final step that we have here is that we are going to recognize the pattern of that object based on its relationship with other geons in that array. So to back up here, again, we recognize that this is a horse compared to a giraffe because we recognize that there's not one geon here that's really, really long, right? If that geon that was really, really long was maybe right here in front of the face, then we might think it's an elephant, right? So it's not just what geons are present in that complex object, it's what relationship do those geons have with each other to help us determine what it is that we are seeing. So, and I feel like I've stuck really close to the data in this chapter, and we're going to continue doing that as we talk about faces as well. But I wanted to show you how Biederman figured this out in the 80s. So he was asking people to say how accurate and how fast, he was interested in how accurate and how fast people were at doing this task. So he was basically like tracking, did they get the question right, and how quickly were they able to make a decision about it. So if I asked you, what is this right here? What would you say? Might take you a second when you're looking at it. It's still a little bit hard, but it is a airplane. What about this? This one, little bit old school, but I'm actually using the same stimuli that Biederman did. This is an ironing board. It's not a complete ironing board because, as you can see, there are some parts missing, right? So there should be another piece that goes out like that, right? For it to be an ironing board. What about this? What is this? That is a camera. What about this? And that's a desk. So basically, what Biederman was up to with this, and you can tell that there was like some weird artifacting going on when I imported this into PowerPoint. Like for some reason, the computer recognized that the back of that tail as an A and just printed an A here. I don't know why I did that. I think it's because the PDF that I was using, I selected recognize text, and it thought that was text. Anyways, thanks computers. Basically, he had 3 different conditions where you had the entire image. That's the complete condition. Then they had where components were deleted out of it, where you can think of this as like, this is some of the geons are removed. And then you had mid-segment deletion, where the geons are preserved, but you had parts in the middle that were erased in some way. And basically, what Biederman hypothesized is that as long as you're able to see the geons, you'll probably be able to recognize the shape of the object or what the object is. If you can't recognize what the geons are, then you'll have a much harder time figuring it out. So, for example, he would suspect that accuracy and reaction time would be best for component deletion, where you have some of those geons present, and worst whenever you delete some of the midsection. So let's take a look if that held up. So here is an example here. They're looking at mean reaction time. So up here, the higher this is, the slower the reaction time. The lower down here, the faster the reaction time. And then he has how quickly he exposed them to those objects. And so you can see that for component deletion, whenever geons were removed, that essentially performance was mostly accurate, mostly the same speed, mostly the same reaction time, no matter how long he showed that image, whereas those in the mid-segment deletion really kind of slow whenever shown for a split second there. So it does change whenever you have mixed segment deletion at these later longer reaction times. And then here, looking at error or the accuracy. So down here, this is kind of a weird way to show it, but down here is actually better accuracy. So because we're looking at error, so higher values would be worse accuracy. I know it's kind of weird, but that's how some cognitive psychologists like to do this, because accuracy is usually really, really high, usually in the 90s and higher. So they'll just plot percentage error, maybe it's a little bit easier to sometimes see differences that way. And so here again, you can see that whenever you're deleting the geons at the 65 millisecond mark, not bad, not shabby. Whenever you're basically seeing it in the blink of an eye, that accuracy is basically, or error, the amount of errors is almost half of what they are making whenever mid-segment deletion is happening. So basically, that was his whole explanation for why geons matter, and people have then gone on to research this stuff for decades and only mostly supported Biedermann's work. So more evidence for Biedermann's recognition by components fall into the visual agnosia. So looking at some of these neuroatypical conditions. Visual agnosia, for example, being whenever you damage the inferotemporal cortex and you're unable to recognize common objects. So your eyes are working just fine, thank you very much. It's just that your brain is not able to put together those geons. It's able to recognize maybe the basic shapes, which is what we see with associative agnosia. So visual agnosia, you can split it up into two separate types. One of those types, you're able to recognize geons, but not able to recognize whenever they are put together. Apperceptive agnosia is just the worst, because then you can't recognize any of these simple objects. All right, let's finish up by talking about face perception. So we're going to be talking about this face or this person or whatever. So do these two faces look different? I'll give you a second here while I take a sip of coffee. Okay, the fun thing about this is that, before I spoil it for you, this is called the Thatcher effect. And it's called the Thatcher effect. This is so weird. It's called the Thatcher effect because it uses an image of Margaret Thatcher. So shout out to any viewers who are across the pond living in jolly old England, the first female prime minister of England. This is Margaret Thatcher, who reigned in the 80s, and it's called the Thatcher effect because it was first demonstrated using an image of her. So you know that one of these faces look different, right? So let's turn them over. Oh my God, what is going on with the Prime Minister? So that's kind of fun, right? But it is very distracting to see where you can clearly tell where they doctored the image, right? You can see where they took a razor blade and just cut it, and so they could flip it over. Times were tough whenever they didn't have computers around, right? This is the kind of stimulus formation you had to rely on. So now we have like Photoshop and can do this research and make it look realistic and seamless. But yeah, now if you're noticing, these lines, you have to forgive them. This was the 80s and this is the best they had. So the whole point of this is that when you're looking at these faces upside down like this, yeah, you can tell that this one over here, that there's something wrong with it. But it doesn't look frightening. It doesn't look scary, right? Whenever you turn it upside down, it looks really scary. It looks really haunting. It looks like, some kind of surgery gone really wrong here. So the reason why it's jarring to us is because the way that we process faces is very different than the way that we process common objects in the brain. I'm going to say that one more time. The way that we process faces in the brain is not the same as how we process other complex objects in the brain. That is processed in a different part of the brain, but also there are certain behavioral things that are not the same. This is one of those examples where when we look at this, we can see that there's a difference, but this difference looks gigantic whenever we have the stimulus right side up. It doesn't look so bad when it's upside down. Same thing with this image. I love this image because those of you that have taken my sensation and perception class, you've seen this image probably already. This is the textbook author of that, and he did a little bit of a better job manipulating his features. You can't really see where somebody took a razor blade and cut around it, right? So which of these faces is the one that's right side up? I'll give you a second to look at it and to guess which of these faces is right side up. If I was measuring your reaction time, it would take you a little bit longer to say what it was. But whenever I turn it upside down, or sorry, when I turn it right side up, it immediately stands out. Oh my God, Dr. Wolf, are you okay? I don't know why you're in a cemetery. That's kind of spooky. But, oh my gosh, what happened to your eyes and to your face, right? It looks like something seriously strange has gone wrong, right? So why is it that these features really stick out to us when it's right side up but not upside down? That is a huge question that psychologists were really interested in answering in the 80s and 90s and early 2000s, and we basically have a good answer now. So if we compare what we saw with this and how eerie it is when it's right side up, compare this. Which of these stimuli is right side up? Which one is correct and which one is incorrect? So one of them is busted. Which one's busted? Now watch as I flip it right side up. Okay, there it is. Didn't even see it, right? And even then, whenever I turn it upside down, it's like I can notice it, but it doesn't haunt your dreams like this does, right? It doesn't feel uncanny. That's a word a lot of people use when they talk about like the uncanny valley. Uncanny valley, man, I wish that's one of those things I wish I could fit into this course. That'd be a really fun thing to talk about. The uncanny valley, like if you think you see a face in a movie and it's mostly CGI, it kind of looks weird and you can notice it's a fake face, right? It kind of freaks you out a little bit where it doesn't feel right. That's the uncanny valley. But we don't get that here when we spin these words around. Right? This one, we know it's wrong, but it doesn't make us feel weird. What about this one? Which one is right side up? This one is kind of weird. You have to think to yourself for a second. And then whenever it's upside down, you can like, okay, yeah, I can see why that one's not right and this one is. But again, it doesn't really stick out to you. So why is that? The reason why is because faces are processed in a different part of the brain. So changes in an inverted face are difficult to detect, but changes to an upright face are extremely easy to detect. The reason for that, as I mentioned, is because faces are not categorized like complex objects in our brain. And we know this not just from behavioral data, like of what I was showing you just now, but because of prosopagnosia, which I mentioned at the very beginning of this chapter. Face blindness, that you can damage a part of your brain called the fusiform face area. We're going to be talking all about it for the next 10 minutes. If you damage that part of your brain, what happens is that you're able to recognize objects totally fine. You're able to say like, yep, these are some headphones. Yes, this is a book. But if my brother walked in the door, I would not recognize who he was. If my wife walked in the door, I would not be able to recognize who she was. So I would not be able to recognize her face. But the amazing thing is that this also means the opposite is true, that for people that have visual agnosia, like we saw, last chapter and just a few minutes ago when we were talking about that, those individuals, they would not be able to recognize that was a book and that was a set of headphones, but they could recognize faces. And so usually that's all they can really make out is faces. There's a really heartwarming part of one of the videos from last chapter where a guy with visual agnosia talks about how all he really has that he can hold on to visually is faces. That's all he's got. That's all he can really recognize, which is really sad, but he's like, I wouldn't trade that up for the other way around, which is really, really hits you in the feels. All right, sorry, that was a very millennial saying. I apologize on behalf of my generation. Okay, so let's get into it. You may remember that we talked about how that there are different areas of the brain that handle different processes, and I said, like, hey, be careful because phrenology did this and it was bad, and so we need to be careful whenever we are talking about specific areas that we're not over extrapolating what they do. So we talked about the O'Craven article, both in chapter 3 and in chapter 2, where if you are looking at this face, and somebody asks you to look at the face, that you're going to have the fusiform face area light up more than if you were told to look at the house here. So the fusiform face area, that's what we're talking about here. The fusiform face area is the area that seems to be responsible for detecting and recognizing faces in the environment around us. And so anytime that there is a face present in the visual field, you're going to see that fusiform face area light up like a firework in your brain scanner. It is one of the most, and I'm exaggerating a little bit, being hyperbolic a little bit, but it is a very, very reliable finding in cognitive neuroscience. In my opinion, it's like one of those that, like I told you in chapter 2, like, yeah, I don't really trust a ton of fMRI data from like the early 90s. But the findings that I think of that really still hold up from the 90s are the ones that look at face recognition, and those that look at the hippocampus. So that look at these areas that are extremely consistent in what they do. The hippocampus, extremely consistent in memory consolidation. The fusiform face area, extremely consistent in perceiving and recognizing faces. It is even active whenever we don't consciously detect a face in front of us. So if, like for example, in this prior slide right here, I don't know if you saw, there were 2 robot faces hiding in the background here. If you had your brain scanned and you didn't detect them, your brain probably would have still have shown some activation of the fusiform face area being active. And that suggests to us that it must be some kind of near automatic process. And then if that's true, then the $1 million question is, well, if we have a part of our brain that is specialized in recognizing faces, does that mean that we're all kind of born with this structure that recognizes faces? Because you can easily imagine the advantages of that, like, right? Like evolutionarily, tons of advantages with that, with being able to recognize a conspecific, being able to recognize somebody of your own species, because that lets you know if they are somebody who's dangerous to you, who might be life-threatening to you, or to let you know this is a potential mate, or to let you know, like, hey, this is a trustworthy person because they are a family member. You can easily see why face recognition would be something extremely valuable in our ancestral background if we're thinking about benefits to survival. But are there any other possible explanations for what this area of the brain could be doing, what it might be up to? And you know that because I posed the question like that there must be something out there, right? So there is maybe another explanation for what this area does. It might not just be for faces. So if it's not, then what the heck is it for? And don't you wish that they told Nancy Canwisher or some of the other, Michael Tarr, or is it Michael Tarr? Yeah, Michael Tarr, or Isabelle Gauthier, these researchers who would go on to do this work. Maybe they should have not called it the fusiform face area, right? if it's not just for faces. So let's take a look at what this area does. I'm going to be talking about Isabelle Gauthier's research here. She did a lot of work with, I believe it was Carnegie Mellon over by outside Chicago, doing a lot of early neuroimaging work in this field to look at the fusiform face area. And so I'm going to talk about her work. And so what she did here is that basically she found that people who were experts in another visual domain also had that area light up whenever people saw instances of that area or of that thing that they're experts in. I know that's kind of vague. I'm speaking in abstracts here. So if I'm being more specific, she brought in car experts and she brought in bird experts, and she scanned their brains while they looked at pictures of cars, while they looked at pictures of birds, and while they looked at pictures of faces, and while they looked at pictures of common objects. So she used the subtraction method in all sorts of complicated ways to try to isolate the activity that was just active whenever people were looking. For example, you take somebody's brain while you're actually, let me draw it out for a second here. So let's say that you scan somebody while they are looking at a face, and you also scan somebody while you have them looking at let's see, this is going to, I was trying to draw like a motorcycle or something like that. I don't think it's going to happen, y'all. I'm sorry. I'm so sorry, chat. Can I redeem myself? Oh, almost. That's like a moped, right? That works. My bad. I should have thought about that a little bit better. It's a moped or a bicycle or whatever. So basically, if you're scanning the brain, oh man, I need to slow down, because even though I know that we're kind of gone on a little long here, I don't want to rush this because I think it'll make sense in a second. So what they're trying to do, they're going to use the subtraction method here. So they're looking at pictures of faces, and let's say that this area is active back here, and this area right here is active, and this area right here is active. And then whenever we scan somebody that is just looking at this right here, you might find this area is active and this area back here is active, but not this area right here. So when you subtract the activation from those two, then what you're left with is just the activation that is unique to that specific task, which would be, in this case, the fusiform face area. Now, they did this exact same thing, but instead of faces, they did this with cars. So they got some, cars. That's definitely a car, right? And then they also did it with birds. And so they're going to take the bird, and then they're going to subtract it from the common object, and then they're going to look at what's left. So the image that I'm going to show you here, that's what they're doing. So they're subtracting faces from objects for car experts here on this row, and for bird experts here on this row. And I know this is a lot of table setting that, I'm prepping a lot of this up, but it's because I think it's really cool if you can interpret these figures along with me. So in this case, we see a little bit of activation over here for the car experts, whenever we subtract objects from face activation. But for birds, we see it's a little bit more robust here. They only use 5 people in these scans, which is crazy, like that they had such a small sample size, and we're still able to get some signal from that. But again, this is one of the things that's been replicated before, so I think it's okay to trust these results. Now they then look at cars and subtract from that common objects. If we look at the birds, right? If we look at the bird experts, we don't really see any new activation. In other words, whenever their brains are looking at cars, it's exactly like when they're looking at common objects. But the car experts, whenever they're looking at cars, they have an increase in activation in their fusiform face area compared to other areas of the brain. And for bird experts, we see just the opposite. So for bird experts, whenever they're looking at birds and we subtract out common objects, the unique activation is going to be in the fusiform face area right there. And that for car experts, to them, their brain does not look any different when they're looking at birds versus when they're looking at complex objects. So, what does this mean? Basically, it means that that maybe part of what people who are experts are responding to when they're looking at something that is in their area of expertise is that area of expertise is handled by the fusiform face area. So if I am somebody who is like, who plays a lot of guitar, and I collect guitars or whatever, and you show me like a 1967 Fender Stratocaster, and I'm like, oh, I know that guitar. It's a 1967 Fender Stratocaster. You know how people can do stuff like that, right? They can do it with cars. They can do it with birds. You might be able to do it with fish or with whatever. That may be the reason why you can do that and why I can do that for specific things, is because we have this kind of this area, this expertise area that is helping us make those visual discriminations. So to test that idea, what Gauthier and colleagues did is they were skeptical of that. They were like, well, we can't say for sure that that's what they're doing. We don't have the data to support that necessarily. So what we're going to do is we are going to make people experts in a field that is totally new, that is totally unfamiliar. And then we're going to see if that new expertise activates the fusiform face area. So because, they didn't assign people as car experts or as bird experts, like they didn't just randomly say like, all right, you're going to be in the car expert group, so spend the next 10 years learning about cars, and you're going to be in the bird expert group, so run along now, spend the next 10 years learning about birds. That basically, she was going to train people to become experts in this new visual domain, and then test the same idea, essentially. So this new thing that they created is called a greeble, and I'm going to show you a greeble. This is what they look like. They look like little demons, like little goblins, right? But they don't really look like anything you can put your finger on. They don't look like anything you can necessarily describe, except this one kind of looks like Dobby, I guess, from Harry Potter, right? But this one also kind of looks like Dobby. They all kind of look like house elves, maybe, that are sticking their tongues out. I don't know. It's hard to describe them using language, and that's part of the point. The other part of the point is that they all kind of look alike, but whenever you put them side by side, you can easily discriminate them. So they are easily discriminable, but not easily recognizable. Okay, so basically what she did is she had people do a pretty common visual recognition task. So it would be like this. If I showed you some of these stimuli and then asked you, do you remember seeing this image? So here you go. Let's do this. Here we go. Here's the first stimulus. Now I'm going to put up a visual mask. Just a quick note about the visual mask, because we talked about the fixation cues already. A visual mask is going to be something usually like this. Like it's either going to be like a gray slide or a checkerboard slide like this, that is going to disrupt any potential visual after images that you might get from looking at a stimulus. So it's a way of kind of like resetting your visual field. That's why people use this stuff, because those things matter when we're talking about cognitive psychology and visual research. All right, so did you see this? Yes, you did. So you'd respond yes, and you get that correct. All right, what about the next one? Did you see this? No, that's new. That's new. That's familiar. That's familiar. That's new. Or sorry, I said that's familiar. That's new. My bad. That's new. Flounder is new. Okay, we've seen flounder before, so we've seen that. We've seen that one before as well. Have not seen that. We have seen that. We have not seen that. Okay. Extremely easy, right? Like that task was pretty straightforward and pretty easy. Now, part of the reason why that was easy is because we have language which can help kind of facilitate that recognition. We're going to talk a lot about that in the upcoming memory chapters and in the concept formation chapters. When we can use language to recall memories, we will. It's just one of the things that our brain does, for most of us. So let's do the same thing now, but for faces. So here we go. So check this person out. She's the first stimulus. All right. Have you seen her? No. Have you seen her? No. Have you seen her? Yes, we have seen her. This is a new face. This isn't a familiar face. This is an old face. We've seen that one. We've seen that one. That's a new one. Now this one we've seen before. We have not seen that one. We have seen that one. We have seen that one. You get the idea. So that one was, again, pretty easy, right? Because we were able to recognize those faces. And if you were playing along, you may have found yourself kind of focusing on, a specific visual item or something, right? And that's okay. Even if it's not exactly how we normally process faces, but we'll talk about that in a second. So now, this is what Gauthier and colleagues did. They used greebles. So have you seen this before, yes or no? All right, so here's the first one. What about this one? Okay, I'm going to go back. Okay, just making sure that is the same, right? It is, but man, it looks, it's that's tough for me. All right, so this one we have seen before. We have not seen this one before. We have not seen that one before. I think we have seen this one before. We have not seen that one. I don't think so. I think we have seen that one. I don't think we've seen that one. We have seen that one. Yes, that one's familiar. I think that one's familiar. You get the idea. So that one was much harder than faces, and it was much harder than the common objects, in part because we did not have language easily around to describe what it is that we saw. And so Gauthier's lab, they've also done this test where they have people learn these names. So this one's called a Doral, and this one's called a Mora, and this one's called a Priya, and this one's called a group. So you learn these words to show that accuracy on a recognition test goes up really high whenever you have words to help with those visual discriminations. If you're wondering what's in the parentheses here, it's a secondary group. Basically learned a separate set of words just to make sure that it wasn't like specific words that were, that were making the distinction easy. Let's do one more time. All right, have you seen this one? What about this one? What about this one? What about this one? This one? This one? And this one's harder, as you can tell, because it is moving around. It's not in the same spot. It's turned over. You have to do some mental rotation. This one is quite hard. So basically, once people were able to do this, like actually perform well on recognizing these greebles, then they looked at their brain activation. So just to look at here, this first one right here, looking at the novices, the greeble novices. So they're doing essentially faces subtracted by common objects, just like you saw before, and greebles subtracted by common objects as well. So this is, for the new folks, that, yeah, the fusiform face area is active whenever you're looking at faces versus any other kind of object. But when they're looking at greebles, no difference. That just like a bird expert looking at a car, like there's no difference between those stimulus classes. However, whenever they train up on greebles, now you can see, like, okay, the face thing is still there, so they still have fusiform face activation. But now, look at all this new activation going on, right? So you have all this new activation after just 5 to 7 days of practicing. So in just that short amount of time, you got people who are able to, or people's brains are starting to respond differently to this stimulus class set. So the long story of all of this is that the fusiform face area, yes, it is used for processing faces, but it seems to be the case that really what it's doing, what the fusiform face area is doing, is it is categorizing and discriminating things that you are an expert in. And it just so happens that as we grow up, because we are social creatures, we spend so much time and energy discriminating faces that all of us are face experts to a degree, because the fusiform face area is always active whenever we come across, faces. But depending on our learning history, if you trained up to be a guitar collector, if you trained up to be somebody who recognizes good visual art and painting, then you can see how that now that you're becoming an expert in it, it's going to also activate the fusiform face area. The alternative, though, I guess you could work the other way, and you could argue that, well, maybe what this means is that now we're perceiving birds like faces, and we're perceiving cars like faces. But I don't know. That doesn't really seem to make as much sense to me. All right. You may remember that when I was talking about the Thatcher effect, one thing I want to make sure that we're all clear on is that part of the reason why that works, why that has such a big effect on us, is because we process faces holistically, which is to say that generally speaking, whenever we are recognizing someone's face, we don't recognize their eye and their nose and their mouth and their ear, and we put them all together. Instead, we recognize how all those pieces fit together. So it's not unlike how we do with geons. You may remember with geons that part of it is like recognizing the basic shapes, but it's also just kind of like being able to recognize the relationship that those geons have with each other. Same is true for faces. That for faces, we recognize what a lot of people will call like the T pattern. So like we recognize essentially like how far apart the eyes are, its relationship with the nose, its relationship with the mouth. And those of you that are art majors or art therapy majors may have some bells ringing in your head, because you might have learned to sketch faces using a technique like this, or probably more likely whenever you're learning to sketch anatomy, you start developing ways of like getting that person's geons together, right? So like before you like sketch out somebody, you have to like draw out, you know, like their outline of these things. I'm doing a really bad job of it right now, by the way, but you get the idea. Then just like you have this outline that has the basic shapes and structures, and then you're going to start filling in the details and so on and so forth. So we see a lot of ties with the visual art world here, like when we're talking about recognizing the way that these shapes fit together. So it's not just recognizing someone's specific eyes, it's recognizing how those eyes fit in relation with the nose and with the mouth and with the rest of the shape of the head. That is what it means for it to be holistic processing. So the inversion effect, which is what we see with Thatcher, is that whenever something is upside down, we have a really hard time discriminating what's wrong with it. But when it's right side up, very easy. That happens when we have stimuli that we process holistically. If we didn't process it holistically, like we did with like some of those words that we were looking at earlier when we flipped them upside down, then that just means that we don't process them holistically. We process them more individualistically. But faces are one of the few things that all of us seem to process holistically. That and also objects of our expertise. All right, that's all that I want to talk about now. Thank you so much for tuning in. If you have any questions, feel free to drop a comment or to email me and let me know. Otherwise, I hope you have an awesome day, and I will see you later. Bye.